Hello everyone, I'm really happy to be here and talk to you this morning about uncovering the secrets of wheat. Are 3 genes better than 1? But before we go into the wonderful world of wheat, I'd just like to introduce myself. So I'm Philippa, and I thought I'd give you a little bit of a background of where I got my interest in plant sciences. So I did my undergraduate degree in Cambridge studying natural sciences, and at that point, before I started my degree, I didn't really think I was very interested in plants. But I had a series of inspirational lecturers, and one of whom's here today, Beverly Glover, who I think spoke to you yesterday. And that's the point at which I really became interested in plants and what they can do for us and for the world around us. So following on from my undergraduate degree, I went to do a PhD at the John Innes Centre, which is in Norwich, which is a research institute, which I'll introduce in the next slide. And during my PhD, I became interested in crops particularly, and I started working on wheat, and that's really the topic that I've stayed working on. And after my PhD, I was fortunate to win a fellowship from BBSLC, which is one of the main UK research funders, and I stayed at the John Innes Centre for that. And subsequent to that, I moved to the University of Birmingham, where I became a lecturer in plant biology. And then I was there for 3 years, and at that point I moved back to the John Innes Centre, where I'm a group leader, where I run my own research group working on wheat genetics and genomics. So a lot of my career has been based at the John Innes Centre, which probably many of you might not have heard of before, because the only thing I'd heard of before was John Innes compost, which actually was invented at the John Innes Centre, but unfortunately we don't make any money out of that, so that was a bit of a missed opportunity. So John Innes Centre is a research institute with a focus on excellence in plant and microbial sciences. We're a strategically funded research institute, and we have 4 main strategic programs of research, one of which is delivering sustainable wheat, which most of my research is focused on. We also have a strategic program in building robustness in crops, which aims to improve the robustness of our crops to climate change. I'm also involved in that one. We have a third program, harnessing biosynthesis for sustainable food and health. And this is more focused on, can we find new biomolecules that will be useful for humans? So for example, on the bottom of the slide, you can see some streptomyces, which are a type of bacteria that we can find new antibiotics and tackle the antimicrobial resistance crisis. And our final strategic program is advancing plant health, where we're trying to improve plant resilience to diseases and pests. And if any of these topics sound interesting to you, we have an international undergraduate summer school, which is open to second year, so penultimate year students. So entries open quite early on. So the next entries will be in January 2025 for summer 2025. So if any of you are interested, I highly recommend you apply. It's I think an 8-week program. It's paid and they give you accommodation. So it's quite a nice opportunity to get research experience. If you Google John Innes Centre Summer School, I'm sure you'll find it. To talk about my research in more detail. So what I wanted to talk about today is why should we care about crop breeding? And then perhaps introducing a different concept of why polyploidy is important. Lots of our crops are polyploids. I'll come back to what that means later on. Why is now a really good time to talk about polyploids? This is something Alessandra alluded to in her introduction with this explosion in new resources for crops like wheat. And then also, how can we unlock polyploidy to maximize crop yields and resilience to climate change? So why care about crop breeding? I'm sure you've all seen some version of this slide before, where the world's population remained pretty much flat until the Industrial Revolution. And since the Industrial Revolution in the 1800s, we've had an explosion in the population of humans around the world. So the current global population is over 6 billion people, and it's projected to reach 9 billion by 2050. And you can see here that the number of people around the world will increase throughout the world. It's not distributed evenly, but this really poses a challenge for us to be able to produce enough crops and enough food for everyone to eat. And we need to find a solution for how to produce these crops. we can't expand the land area, there isn't more farmland available, so we need to improve the yields of crops that we have. But we also need to carry this out under the increasing changes in our weather and climate that we see due to climate change. And this really means we have a great challenge and an opportunity in plant sciences to really make a difference to the world. This might sound like a really daunting task, but we do have a historical precedent that this could well be possible. So if we go back to the before 1950 really, so this is a painting by Peter Bruegel the Elder called The Harvesters. What I want you to notice is, hopefully I can use you can see this man in the middle, and he's standing in a field of wheat, and you can see that the wheat is about the height of his head. So historically, wheat plants and other cereal crops were really tall. And this was a bit of a problem because they tended, if it was windy, they would fall over, and that could ruin the grain. So by the time we get to the 1960s and 70s, this is again a field of wheat, and you can see a dramatic reduction in the height of these wheat plants. So it's kind of waist height now. And this is due to the Green Revolution that happened from the 1960s onwards, which was really led by plant breeders. And this enabled a huge increase in yields, as you can see in the red line, without increasing the area on which crops were grown. And this was really effective in more than doubling yields in many parts of the world. These are just a few examples, so USA, China and India, but it wasn't equally felt throughout the world. So for example, in Africa, the impacts were much more limited, so there's still work to be done. And this crop revolution was really focused on grain crops. So for this I mean wheat, maize and rice, which are the most important crops and have the largest production globally. And this green revolution was achieved by several different methods, the first of which was shuttle breeding. So this was a new innovation that rather than growing crops once per year, they used two different climates within Mexico, the Yaqui Valley and near Chapingo, and then they could grow 2 crops per year. So that meant in 1 year you could do twice as much plant breeding as you could before. A second innovation was introducing multiple disease resistances together in the same line. So this meant if you had a disease and it overcame one particular gene by evolution, then you'd have other backup genes available which could still resist the disease, and that made them much more effective. The kind of most obvious change you could see from this green revolution was the reduction in plant height, as I mentioned earlier. So this was achieved by introducing reduced height genes in wheat from a variety called Norin 10 from Japan. And on the right, you can see the very short variety, Norin 10. On the left, you can see the traditional varieties. When you cross them together, you can make an intermediate, which has this waist height size, which is really beneficial. And the main advantage of this is that by reducing the height, more of the mass of the plant goes into the grain, so you don't waste lots of energy from photosynthesis in the non-edible straw. More of what you harvest is actually grain that can be eaten. And this also had the advantage that these plants were less likely to fall over in high winds, so more of the crop could be harvested, and it allowed the application of fertilizers as well. If you apply fertilizers to the really tall varieties, they just grow even more and just all fall over. And the final thing was the use of a method called the pedigree method, which the details aren't so important right now, but the important thing is you cross together 2 different parent plants, and then you segregate out the offspring, and you pick the best ones and go through multiple generations, picking the best one each time until you have an improved variety. And you can then repeat this, crossing the best with the best to produce even better lines. So this green revolution was largely led by Norman Borlaug, and in 1970 he was awarded the Nobel Peace Prize for his major impact on global peace because of this huge increase in crop production, around more than 2 times in many parts of the world. So we've done it once before, but we still need to do it again. So plant breeding provided this step change from the 1960s onwards, and it was really focused on cereals like wheat, maize and rice. And now I think we really face a challenge to be able to produce another step change in crop breeding. And today I'd like to tell you about why I think polyploidy could be one route to unlock this step change. So why is polyploidy important? So polyploid plants are plants that have had a whole genome duplication, so they have multiple copies of the genome. And I'll give an example of wheat how this comes to happen. And you might be thinking, I don't really know any polyploid plants. But Polyploid plants are incredibly common amongst our crop plants. So over 70% of flowering plants, so many of the plants you see growing around you, have been polyploid at some point during their evolutionary history. And many of our major crop plants are polyploid. So these are just a few examples. So we've got oilseed rape, cotton, potatoes, bananas, sugarcane, and wheat, which is my favorite polyploid. So probably most of you for breakfast have eaten something from a polyploid. Maybe you had some toast or some other kind of cereal made from oats, also a polyploid. So these are really common polyploids in plants. They're less common in the animal kingdom, but because polyploidy has affected so many of our major crops, we need to understand how this process happens and what the implications are for plant growth and particularly crop production. So I'll be focusing on wheat, which is a major polyploid crop. It provides about 20% of calories eaten by people globally, which is a really astounding fact when you think about it. So a fifth of everything that people eat is wheat. So wheat evolved from several diploid ancestors. So these are plants that aren't polyploid. So they just, like humans, have one set of each chromosome. So two different diploid ancestors, Triticum murata and a B genome progenitor, which is now thought to be extinct, but was probably something similar to this Aegilops speltoides, hybridized around 400,000 years ago. And during this hybridization, both genomes were retained. So the A genome from Triticum murata and the B genome from the ancestor thought to be similar to Aegilops speltoides. And this means that we now have a tetraploid. So we have, instead of just having the A genome and the B genome, now we have both combined together within one species. But this doesn't make it complicated enough. So in wheat, it's actually a hexaploid. So we have another step of hybridization. So another diploid grass species called Aegilops tauschii, which has the D genome, hybridized again to this tetraploid about 10,000 years ago to produce hexaploid wheat. So again, all the genomes were retained. So now, instead of having started from a diploid with just one single genome, we now have three subgenomes, the A, the B, and the D in the wheat genome. This isn't the only challenge associated with working with wheat. So we've got these three genomes, the A, the B, and the D. They all came from different wild grasses, but they were quite similar to begin with. So they're fairly similar across the whole genome, but with slight differences, hence my colouring them as kind of shades of red. But wheat also has a really large genome. So to put it into scale, these are the sizes of some other, humans. So wheat genome is about 5 times bigger than the human genome, but also it's much bigger than other plant species that are commonly used as model species like Arabidopsis and rice. And if I put something like E. coli, which is a bacteria used for lots of experiments, it would be pretty much the size of a dot and you wouldn't be able to see it on the slide. So this huge size of the wheat genome has made working with wheat a bit more tricky, especially in the genomics era, where we like to be able to obtain the entire genome sequence of organisms that we work with, to be able to target them in a really gene-specific manner. But we are getting there in wheat now. So this is what the wheat genome and chromosomes looks like. So again, we have the A, the B, and the D. And because these three grass species were quite similar to begin with, if we have a gene on, say, chromosome 7A, we will likely have a very similar gene on chromosome 7B and a very similar gene on chromosome 7D. And that's because the three different grass ancestor species were quite similar to each other to begin with, so they had quite a similar set of genes. So we can find these kind of matching genes across the three subgenomes. And I'll be talking about these three genes quite a lot in my talk. And these are called homeeologs. So homeeologs just means the three gene copies in wheat between the A, the B, and the D subgenome. And these homeeologs are very interesting because they're quite similar in their sequence. They're over 95% similar in their coding sequence. This is the part of the gene that makes the proteins that then carry out all the functions within the cell. So one of the key questions I'm interested in my research is, are these three homeeologs the same or different in terms of their biological function? They're more than 95% similar, but there is some difference. So is that important? And how does that affect crop breeding and what we can do with wheat? So we know that these wheat gene copies can act in different ways. So if we take a step back and move to a diploid like rice, so rice only has one set of each gene, just like we do as humans. And in rice, if we knock out a particular gene called grain width 2 that affects the width of the rice grain, you can see that by knocking it out, you get a huge increase in grain width. If we do the same thing in rice using the orthologous gene, this is the same gene but in a different species, then in wheat, if we have the wild type, so the normal plants at the top, we can knock out a single copy, we get a bit of an increase. We can knock out 2 copies, we get another increase, and knock out all 3 copies, we get an even bigger increase. So we can see we have this additive effect of the 3 gene copies. And so this cumulative effect can sometimes be seen in wheat. So we have some genes that act in this way where knocking out 1 copy, you get a slight increase, 2 copies, a bigger increase, 3 copies, you get a really big increase, which is much more comparable to what we would see in a diploid species like rice. But that isn't the only way that genes can operate in wheat. Another way is that they act dominantly. So in this case, just 1 copy, if you change it, has a really strong effect, just like as if it was a diploid. So it seems like in this case, the other 2 gene copies don't really matter. So this is what I will call dominant. But we also have cases where the 3 copies are redundant. And what I mean by this is if you knock out 1 copy, nothing happens. In this example, the grain would stay really thin. If you knock out 2 copies, they also would stay really thin and nothing happens. So you might think, okay, this gene has nothing to do with grain width. But when you knock out all 3 copies, you suddenly get an increase in grain width. And this is because the 3 copies can compensate for each other, so they're acting in a redundant manner. These kind of different ways in which the copies can work means there's a lot of potential in the genome that we haven't been able to study until recently. So just to show one example, here we've got a gene that's redundant between the 3 copies. This is a starch branching enzyme. So starch is the main component within the grain that we associate with getting calories from these carbohydrates in our diet. And starch is made of 2 main components called amylose and amylopectin. And if we look at a normal wild-type plant, which hasn't got any mutations in this gene, we can set the percentage of amylose content in the grain to be 100%. And if we knock out either 1 or 2 copies of this starch branching enzyme 2A gene, we don't change amylose content in the grain. It's exactly the same as wild-type. But when we knock out all 3 copies, we more than double the amount of amylose within the grain, showing this gene is really important for affecting the amount of amylose within the grain. And so if we can more than double this particular trait, then maybe we can more than double other traits. Maybe we could more than double, disease resistance or the size of the grain or many other traits that we might be interested in for agriculture. But really the problem we face at the moment is that in wheat, we don't know which gene acts in which way. So we know that some genes act in these different fashions, but we can't say just from looking at their sequences which ones are going to be cumulative, which ones will be redundant or dominant. And this would be really useful because if we knew that a gene was, say, dominant, if you're a crop breeder, you should say, okay, well, I'll just look at one of them and focus my breeding efforts on that one because that will give me the strongest effect. That's all I need. But if we had genes which are redundant and you just looked at one, nothing would happen and that would be completely useless. So at the moment, we have to hedge our bets, really, and focus on all 3 copies most of the time. But hopefully in the future, we'll have a better understanding of which genes operate in which manner, and therefore be able to take a more targeted approach, and for at least some genes, save ourselves 2/3 of the effort by just looking at the one gene, which is the most important. So why do we need to understand polyploidy? And I would say that polyploidy is really common in our major crop species, and it also hides a lot of variation. So we have these redundancies between gene copies, which means that by just changing one gene copy, you don't really see any change to the characteristics of the plant due to compensation by the other copies. But if we could actually unlock this variation, we can make huge changes to the characteristics of crops like wheat, and therefore be able to solve some of our problems in terms of producing crop production and also combating climate change. So why now? So these problems have existed for a long time. Why is now a time when we can realistically start to tackle them? So for wheat in 2018, we completed a high quality chromosome level genome sequence for wheat. We were able to assemble over 94% of this huge genome. And we were also importantly able to arrange it into pseudomolecules, which you can think of as being chromosomes. So we're able to arrange the whole genome into actually nearly entire chromosomes. And this is important because before we had some genome sequences for wheat, but they were a bit more like a jigsaw puzzle that hadn't been assembled. So we had all the pieces, but we didn't know how they all fitted together. But now that we have this genome sequence, which is highly contiguous, so very well assembled, then we can actually put together most of these jigsaw pieces. We have a picture of the whole puzzle. And that means we can really start understanding what's going on, because if you have a jigsaw piece that's just a blue bit of the sky, it's not very helpful. But if you now know exactly where it is in the sky, and you know it's next to a cloud, or then it can be much more informative, and you can actually use that for crop breeding. And also having this whole genome sequence meant for the first time we could understand how many genes there were in the wheat genome. So there are about 110,000 genes in the wheat genome, which is more than 3 times the amount of genes in a human genome. So wheat is a really complex organism. But as I mentioned, this is because it has this 3 copies of most genes. So that means you have kind of duplicates of many of the genes, and this means we have some challenges due to redundancy, but also opportunities to enhance the variation we can see naturally. And wheat isn't the only plant that's been sequenced now. So there's been a huge increase in the number of plant genomes sequenced. So the first plant genome sequence was Arabidopsis in the year 2000, and since then you can see this dramatic increase in the green bar. As time goes on now, thousands of genomes are being sequenced per year. And these are also increasing in their quality. So you can see that to begin with, a couple were chromosome level. Then we went through a long period where we could sequence genomes, but we couldn't really assemble them fully, so they weren't chromosome level. But now most of the genomes being sequenced are increasingly chromosome level. So they're, instead of being individual pieces of the jigsaw puzzle, they're the entire jigsaw already put together, ready for researchers to use. So this means that we can actually start trying to find out which of these genes in wheat act in which way. So hopefully we can discover whether we should target individual genes, so we target just one copy of a gene, or do we need to target multiple copies for crop breeding? So as the first step, we decided to study gene activities, so specifically looking at gene expression. So this is whether the gene is switched on or switched off. And we can think about it as similar to lamps in a room. So you've got the 3 lamps, they're in your room, but they may not be switched on at the same time. And the same is true for the genes in the wheat genome. So within every cell of a wheat plant, all 3 genes are present, but they might not be active, so they might not be switched on. So that means that each cell could be different. And we wanted to know what's actually happening. Is it that always all 3 genes are switched on, or are there differences between these gene copies already at the level of gene expression? So we could have a case where 1 copy is switched on, 2 copies are switched on, or 3 copies are switched on. And until having this genome sequence, we couldn't understand how many genes fit into each of these different categories. So using RNA sequencing, we studied how the genes are expressed in each of these categories across the whole wheat genome. And what we found was it was quite rare to just have 1 of the 3 genes switched on. So around 2% of the time, then just say the A homeolog would be switched on, or the B homeolog, or the D homeolog. It was also fairly rare, but a bit more common, that 2 out of the 3 would be switched on. So here you can see it's quite even across the 3. We didn't find that 1 of the 3 copies was more common to be switched on. In some polyploid species, it is the case that 1 of the subgenomes is dominant, so it's more active. But in wheat, it seems very even between the 3 subgenomes at a global level. And we also found that the majority of genes have all 3 copies switched on, and they're expressed at a balanced level, so all 3 copies are expressed to a similar degree. So this was 72% of genes across the genome. So that was interesting, but one thing we also noticed was that actually this isn't the same for all different varieties of wheat. So wheat has different varieties, which are like different breeds, if you like. So if you think of dogs, they're all the same species, but they look very different from each other. We have the same thing in wheat. So we have different varieties which might be used for different purposes. So one might be used to make bread, and one might be used to make pasta. And we can look Sorry, that was a bad example. Pasta wheat's actually a tetraploid, so scrap that. Hope you can take that out of the recording. So you have different breeds of wheat, and one might be very tall, one might be very short, one might be used for making bread, one might be used for making cakes. And these different breeds of wheat also have differences in these expression patterns, which we've discovered. So for example, here we've got a nominally blue wheat variety and a nominally orange. They're not really different colours, they're just for illustrations. And we notice that some of these genes have different activities between them. So for example, the blue one, the B copy is more highly expressed, but in the orange one, the A copy is more highly expressed. And something that we've been working on recently with two members of my team, Marek and Arun, is what happens if you cross together these different wheat varieties that have a different expression pattern. So wheat breeding and plant breeding, as I mentioned earlier, is really based on making crosses between different parental varieties and then looking at the offspring and picking the best ones. So what's happening when we're actually crossing together different wheat varieties that have these different expression patterns? So we've looked at this. We crossed together two different wheat varieties and looked down to the 6th generation where everything should have become stable again. We won't have too much. Each line will become more fixed and stable. So we could get examples where one of these offspring looks very similar to one of the parents. So in this case, we have this line 1, and it pretty much looks identical to the blue parent. And that seems quite logical. It's inherited all these A, B, and D copies from the blue parent, and so the expression level looks very similar to the blue parent. We can also get cases where the A, the B, and the D are inherited from different parents. So if you look particularly at line 2, you can see that the A copy is reasonably highly expressed, and it's inherited that copy from this orange parent that had quite high expression of the A. Okay. Then we've got the B copy where, again, it's reasonably highly expressed, and that's from the blue parent. And okay, the B was quite highly expressed, that makes sense. And then the D is a bit more low expressed, and that came from the blue parent again, and that was a bit lower expressed there. So that seems all to make sense. And then we also have the same case in line 3, so high in the A because it's from the orange parent, and then reasonable expression in the D from the blue parent. But we also noticed you get completely unexpected expression patterns. So here in this case, this 4th offspring that we looked at, then the A was inherited from the blue parent, but in the blue parent it really didn't get expressed, but now in this offspring it's very highly expressed. So we can see new patterns of activity happen during crossing, and this could potentially really alter the characteristics of the plant, and that's something that we're going to be looking into more in the future. So why now? Why is it now a good time to investigate polyploid crops? So for wheat specifically, we have a fully sequenced wheat genome, which allows us to really start exploring the function of individual genes. And we've discovered that many homoeologs, these 3 gene copies, are expressed in unbalanced ways. So about 30% of the time, the A, the B, and the D are not expressed to the same level. And this could be the first hint that they're not really redundant. So if one is more highly expressed, then maybe that's the one that's important that we should focus on our breeding efforts. But that's something that still needs to be confirmed experimentally. So in the final part of my talk, I wanted to talk about how can we unlock polyploidy. So we've got these 30% of genes that potentially are not redundant, but we've still got to deal with the 70% of them that the 3 are expressed in a very similar way and therefore may be redundant with each other. So how can we start to understand the roles of these genes and use them in crop breeding? So I want to focus on one particular trait that we're interested in my group, which is the trait of senescence. This is actually the perfect time of year to talk about senescence for wheat, because you can see it happening, or we will see it happening in the next couple of weeks in the fields around. So if we look at this aerial view of some wheat plots, we can see that over time the leaves turn from green through to brown, and that's the process of senescence. And senescence is really important because it causes an influence on both the yield and quality of the grain. And that's because during this time, the plant has some decisions to make. So if the plant was to stay green for longer, so it delays senescence, it could potentially do more photosynthesis, fix more carbon, which could then be transferred into the grain and potentially increase the yield. But the problem with that is that then the leaves are staying green for longer. Lots of the nutrients, particularly nitrogen, will stay in the leaves. That means that they aren't moving into the grain, so the quality of the grain could go down. And we could have the inverse situation where the plant senesces more rapidly. This could allow nutrients to be remobilized, like nitrogen. That could increase the quality, particularly the protein content, which is really important for many of the baking properties of wheat. So if you want to make a good loaf of bread, you need to have high protein content. But the problem with this, you've senesced rapidly, so that's decreased the amount of photosynthesis the plant could do, and that might have a negative impact on yield. So getting this balance right is really important for the final properties of the grain and what they'll be used for. And it's also important for tailoring the plants to the environment in which they'll grow. If you have a shorter growing season, you might want to have a more rapid senescence, for example. So this process of senescence is really important for both the yield and the quality. And it's controlled genetically by multiple genes. But in wheat, until recently, this was really quite a black box. We didn't know which genes were involved in controlling senescence, so we couldn't make tailored changes to the senescence using a gene-by-gene approach. And this was in part due to the polyploid genome of wheat. So in many cases in plant science, if we have a trait that we're interested in, and we want to understand which genes control that trait, we'll carry out a mutant screen. So we'll take a whole load of seeds, add a mutagen, something like a chemical that can change DNA, and then screen that population for changes to our trait of interest. So we have tried this in senescence in wheat, and others have tried this too, but the issue is you only find really one gene that's influencing senescence, and that's, I don't think it's because one gene is in control of everything during senescence in wheat. Instead, it's due to this redundancy problem. Because if you do a random mutagenesis, by chance, you're only going to hit one of the 3 gene copies. It's extremely unlikely you're going to hit all 3 gene copies using random approach. And that means if you've knocked out one copy of a gene that affects senescence, you'll never see it because it will look exactly like a wild-type plant in your mutant screen. So this has made it quite difficult to understand all the different genetic components that control senescence. So we decided to take an alternative approach, and this was using an approach called gene regulatory network modeling. In essence, what we can do is pick apart the effects of the individual homeologs and then identify genes that we couldn't find otherwise. So to do this, we used RNA sequencing, which measures the gene expression or activity of the genes. And we did this across a time course. So we can measure gene expression across time. And then we can use this information about gene activity to build a network of which gene is influencing which other gene. So in this example that you can see at the top, we just have quite a simple network. And you can see there's the blue gene in the middle that's connected to many other genes. So we'd expect that this gene is really central in the network. And if we took that gene out, for example, through a mutant, we'd see a strong effect on senescence. Luckily in wheat, we now have a sequenced collection of mutants, so we can just go to a catalogue, say I want a mutant in that gene, and we can order it and then grow those seeds and see what the effect on phenotype is. And what we're hoping to see is a strong effect on senescence. So when you knock out the gene, you might have a much later senescence, so the plants will stay green. And when you keep the gene, you have a more rapid senescence, like you can see on the right. So this is exactly what we did. So we looked at an early time course of senescence. So this is from 3 to 26 days after anthesis or flowering. And what you can see here are photos of the leaves of wheat that we looked at. This is the flag leaf, which is the top leaf of the wheat plant. And we picked that one because it provides about 50% of the photosynthate to the developing grain and more than 50% of the nitrogen to the developing grain. So it's a really important leaf for this balance between yield and quality. And we decided to look at quite an early time course of senescence. So even by our final time point, you can see the leaves are starting to go yellow, but they're still mostly green. And this is because we hypothesized that if we want to identify the strongest effects on senescence, we should look early in the process where we'll have the most dramatic change to senescence if we alter the genes involved in that process. So we did RNA sequencing at each of these 10 time points, and we're able to use this information to build a network. So there won't be a quiz about the network, you'll be relieved to hear. But the idea is that each circle represents a gene, and the lines represent the interactions between the genes. And this network does have a directionality. It goes from the bottom to the top of the screen. And they've also made the genes sized according to how many connections they have. So you can see there are some genes which have a lot of connections because they're really big, and some that don't have that many connections, so they're quite small. So this was nice. We had a network with around 300 genes in it, but was it telling us anything actually useful about senescence? So we first of all wanted to check for a gene, the one gene I mentioned earlier that we do know influences senescence in wheat. And this gene is called NAM. And luckily we did find the NAM gene in our network, so that really validated that our approach was working. So we talked about earlier how wheat has 3 gene copies of most genes. This one actually has another duplication to another chromosome, so that's why we've got the 4 copies at the bottom. And these were quite near to the start of the network, and that also fits with our understanding of this gene's function, so it has a really strong effect on senescence. But we wanted to find new genes, and something we noticed straight away was there was actually an additional copy of this NAM gene called NAMA2 that hadn't been previously identified or characterized. And this was because when this gene was cloned back in 2006, there was no sequenced genome for wheat. So this gene just hadn't been found because no one knew it was there. So we thought, okay, we have this extra gene copy, its close relatives are involved in regulating senescence, that will be a great one to start with. So to do this, we took a jump into tetraploid wheat. So this one is a bit more simple. We just have 2 gene copies. This is really for practical reasons to save time, because we need to cross together mutants in each gene copy to make sure we've completely knocked out the gene. So in this tetraploid wheat, we have the A and the B subgenome. We could find mutants in the A that would knock out this gene, mutants in the B, and then we'd cross them together, self those plants, and then we'd get a segregation of some wild-type plants on the left in the big letters, and some complete mutant plants on the right with the lowercase letters. And we used this to test, was NAMA2 involved in regulating senescence? And this is what we could see. So in our wild-type plants, the leaves have gone completely yellow. In the single mutants, you maybe see a little bit more green, but then in the double mutant, the leaf is much more green. And when we quantify this, it is a statistically significant difference in the flag leaf senescence rate. And also we saw changes in the peduncle, which is the part of the stem that connects together the leaf to the grain. And the timing at which this peduncle tissue senesces is really critical, because once it's dead, obviously no more nutrients can go through into the developing grain. So that was great. We'd found a new gene, but it wasn't new. It was quite similar to some other genes we already knew. So we wanted to say, can we actually find completely novel genes that we couldn't have found through any other approach? So we decided to focus on the NAC transcription factors, which are relatives of these NAM genes. So transcription factors are genes that control the expression of other genes. And the NAC transcription factor family is unique to plants, and it's known to be involved in regulating many different biological processes. So it regulates senescence, but it also affects things like abiotic and biotic stresses, so responses to, say, drought or heat, and also responses to pathogens and diseases. So we've got lots of genes in the wheat genome. There are hundreds of NAC transcription factors. In our network, we had about 50 that we predicted were involved in senescence. So we decided to start characterizing these genes, and I'll just show one example. So here is a NAC called NAC5. And Catherine, a PhD student in my group, characterized NAC5. And what she found was, again, using the same approach, where we use a tetraploid wheat to speed things up. When we knock out NAC5, we can see the plant stays green compared to its sibling that has a functional copy of NAC5. And we could quantify this again. So in this case, the mutants in blue have a higher amount of chlorophyll within the leaves. This is the green pigment. So that suggests that they're senescing slower. And we also used the transgenic approach to overexpress NAC5, and we found the opposite. So when we overexpress NAC5, we get an acceleration in senescence. So a reduction in chlorophyll content in the flag leaves. So that shows that really our network can actually predict new genes involved in regulating senescence, and we're currently working to characterize other genes within that network. So we can use RNA sequencing to tease apart these effects of the gene copies that we couldn't understand using approaches like mutagenesis. We can build a gene network in wheat, which is really underpinned by these new genome sequences that we have available for wheat. And then we can predict which genes might be involved in regulating a particular trait that we're interested in our case, senescence. We can use sequence mutants that allow us to just rapidly order a mutant in our gene of interest. We still have to cross them together, so that does take a bit of time, but it's much quicker than previous methods. And then we can take these mutants into the field to test what they do really in the field. So the data I showed you was just from greenhouse experiments, but we're working closely with several wheat breeding companies to translate these findings, move our plants out of the greenhouse and into the field, and not just measure senescence, but also look at the effects of these mutants on yield and protein content. So in conclusion, I've talked to you about 4 main questions today. So why should we care about crop breeding? So we really need to increase food production, and we also need to do this under the threat of climate change and be able to produce crops that can cope with increasingly unpredictable weather conditions. And I think that we need to understand polyploidy because these crops, which are widely eaten around the world, have to be part of that solution. They're really widespread, and they also have been historically quite hard to work with, but I think they represent a great opportunity because we have a lot of diversity within their genomes that's currently hidden by redundancy. So by understanding which genes are doing what, we could start to unlock this variation and these new characteristics or increased variation within characteristics that we want in our crops. I think now is the perfect time, so we have new genomic resources for wheat and also for many other polyploid crops. And we've been able to take the first steps towards understanding redundancy between homoeologs. And we found that about 70% of the time, then the 3 copies are expressed in a similar level, but 30% of the time, the expression levels or activities of these genes differs, which might suggest they are not redundant completely, and that could present a new opportunity for crop breeding, where we only need to look at 1 copy and not worry about the other 2, so that would cut out 2/3 of the work for a plant breeder. And finally, I showed you one approach we're using to try to unlock the challenges associated with polyploidy. So we've been using gene networks, using RNA sequencing to identify new genes involved in regulating senescence and understand how we can improve both protein and yield within wheat. So I'd like to end by thanking everyone in my group who's contributed. I tried to highlight the individuals involved as we went along, but that was particularly Catherine, Marek and Arun. I'd also like to thank our collaborators, so Cristobal Wowie at the John Innes Centre and many other people in his group, particularly Ricardo, with whom we did the work looking at gene expression levels across the genome to start understanding redundancy, and also the whole of the International Wheat Genome Sequencing Consortium, without whom this wouldn't have been possible, because without a wheat genome sequence, it would really be impossible to carry out this work. And this was a project that went on for almost 20 years to be able to sequence the wheat genome. So it's really been a huge international effort with people from more than 60 different countries. And that was really an exciting project to be involved in. And finally, I'd like to thank all our funders and you for your attention. Thank you.